Hello everyone and welcome to K Foundations. Myself Uthara, and today we'll be starting Cell the Unit of Life chapter, which is chapter 8. Okay, so let's begin. When you look around, you see both living and non living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have, which a living thing has? The answer to this is the presence of the basic unit of life, which is a cell, right? D- um, the cell distinguishes between a living and a non-living organism. All organisms are composed of cells. Some are composed of a single cell, they are called as unicellular organisms, and some are composed of many cells, which is multicellular organisms, right? Uni and multi, one and many. Okay, so what is a cell? Unicellular organisms are capable of important independent existence and performing the essential functions of life, right? Anything less than a complete cell, does not ensure independent living. For the independent existence of living organisms, anything less than a complete structure of a cell, right? If the cell is losing any part of its components, then it cannot ensure that it can live independently. Okay, hence cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Okay, he described a live cell, while dead cell, basically dead cell, um, or the cell wall, was observed by Robert Hooke. Right, Robert Hooke had observed the cell wall or a dead cell, right, a coxal. Okay, so Robert Brown later discovered the nucleus, right? The invention of the microscope and its improvement leading to the electron microscope had revealed the structural details of the cell. The structural details were revealed by electron microscope, right? Yeah. Moving to the cell theory, in 1838, Matthias Kleeden, a German botanist, had examined a large number of plants and observed that all plants are composed of different kinds of cells which forms tissues of all of the plant. At the same time, Theodore Schwann in the year of 1839, a British zoologist, right? Remember the point that 1838, 1839. Matthias Kleeden, Theodor Schwann, he was a German botanist, British zoologist. See, botany and zoology, Matthias Kleeden and Schwann. So Matthias Kleeden, Theodor Schwann, 1838, 1839. So he was a botanist. He spoke about the plants, okay? And now Theodor Schwann was a British zoologist. Okay. He said that he studied different types of animal cells and reported that cells had a thin outer layer. Okay, it formed a thin outer layer in animal cells, which is today known as the plasma membrane. Okay, so he had observed a plasma membrane, of course, right? Because animal cells don't have cell wall. He also concluded, based on the studies on plant tissues, that the presence of cell wall is a unique character of the plant cells. Okay, presence of a cell wall is a unique feature of the plant cells. So you shouldn't guess it like in the plant, right? Plants will be studied under botanists, so it was given by Schleiden. No, it was given by Schwann itself. Okay. So first point is he spoke about plasma membrane. Second point is he spoke about cell wall of the plant cells. And finally, he proposed a hypothesis that the bodies of animals and plant cells are composed of cells and the product of cells. Okay. Yeah. Schleiden and Schwann together formulated the cell theory. Okay, this theory, however, did not explain how new cells were formed. It did not explain how the new cells were formed. Okay, so Rudolf Virchow in the year of 1855 had explained that cells divide and new cells are formed from the pre-existing cells. This is called as omnicellular e cellula. We know, right? The pre-existing cells are going to divide and it's going to lead to the formation of the new cells or the daughter cells. Yeah. He modified the hypothesis of Schleiden and Schwann to give the cell theory a final shape. Okay, so it was that all living organisms are composed of cells and the product of cells, and all cells arise from the pre-existing cells. Okay, so Rudolf Virchow, in which year he had modified the cell theory? 1855, right? So Matthias Claden, which year? 1838, and 1839 was Theodor Schwann. Okay. Next, an overview of cell. You have earlier observed cells in an onion peel or a human cheek cell under the microscope. Let us recollect their structures. The onion cell, if you see, it's a typical plant cell because it has a distinct cell wall as its boundary, and just within it is the cell membrane, right? Because onion cell is a plant cell, it has a cell wall, so it's distinctive, and you can see the cell membrane just within the cell wall. The cells of the human cheek has an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane bound structure called as the nucleus. Nucleus is membrane bounded. Okay. This nucleus contains the chromosomes, which in turn contains the genetic material, which is DNA, right? So you can see the nucleus. Within the nucleus, you can find chromosomes, and in the chromosomes, within the chromatids, you can find the DNA, which has the genetic material. Okay. Cells that have membrane bound nucleus, membrane bound nuclei in the sense eukaryote, okay, and cells that lack a membrane bound nucleus is about the prokaryotes. Okay, so you should see that if the organelles and the nucleus is membrane bound, then you can find them in eukaryote, and a lack in the membrane bound nucleus is a prokaryotic organism. Okay. Yeah. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called as the cytoplasm occupies the volume of the cell. So I am sure you'll be aware of them, right? So the cytoplasm is the main arena of all the cellularic activities in the plant and the animal cell. Various chemical reactions also occur to keep the cell in a living state. Okay, speaking about the main arena of all the cellularic activities, if you see, it'll be the cytoplasm, right? In the cytoplasm, you can find various cell organelles, and within them is the reaction occurring. So it's indirectly in the cell cytoplasm, right? Hmm. Besides the nucleus, the eukaryotic cells have other membrane-bound distinctive organ structures called as the organelles. You can find endoplasmic reticulum, Golgi, lysosomes, mitochondria, microbodies, vacuoles. The prokaryotic cells lack such membrane-bound organelles because in prokaryotic, it's basically a primitive cell type, okay, where you can't find such membrane-bound organelles. But like it's not there are certain exceptions, like as in ribosomes, they are non-membranous, but they are found in both prokaryotic as well as eukaryotic. Okay, and within the cells, ribosomes are found, yeah, where you can find ribosomes are in mitochondria, chloroplast, right, rough endoplasmic reticulum, okay, and finally in the cytoplasm. These four are the sites where you can find our ribosomes. Okay, within the cells, ribosomes are found not only in the cytoplasm, that means they are in the cytoplasm. Along with that, they are in the chloroplast, mitochondria, and the rough endoplasmic reticulum. Animal cells, yeah, this is important. Animal cells contain another non-membrane bound organelle called as the centrosome, which helps in cell division. Next chapter will be about that only, right? So to be remembered that centrosomes are only in the animal cells, they are not in the plant cells, and also they lack membrane. Okay, now cells differ greatly in the size, shape, and activities. For example, mycoplasmas, talking about them, they're also called as PPLO, right? Pleuropneumonia like organisms. So they are the smallest cells, okay, only zero point 0.3 micrometers in length. While talking about bacteria, there'll be 3 to 5 micrometers in length. We'll come to the diagram. 3 to 5. And the largest, first of all, human RBCs are 7 micrometers in diameter. Okay, so the largest isolated single cell is the egg of an ostrich, and nerve cells are the longest cells. Okay, largest isolated single cell, egg of an ostrich, and nerve cells are some of the longest cells. Cells can vary greatly in their shape. They might be disc like, polygonal, columnar, cuboid, thread-like, or even irregular. The shapes vary with their functions. Shapes vary with the functions they perform. Okay. Yeah. So to remember the numbers, just write 0.3 micrometers. This 3 you can write it back, 3 to 5 micrometers. Okay. And then 5 plus 2, 7 micrometers. PPLO. Okay. Bacteria. Human RBC. Okay. Talking about the diagram, if you see, yeah, RBCs are round and biconcave, correct? Yeah. So they are round and biconcave, and they are called as erythrocytes, right? And WBCs are amoeboid. What is the reason for the shape? Because they have to they have to perform diapedesis, right? So that's the thing, and they are also called as leukocytes. And that is columnar epithelial cells, long and narrow. Columnar epithelial cells are found in the last type, right? So you can find it in stomach, intestine, and all. If they bear cilia, then they'll be ciliated columnar epithelium, right? Nerve cells, they are branched and long, correct? Yeah. And here you can see the trachea, it's an elongated type. Mesophyll cells are round and oval. Okay. Yeah. So now moving to the prokaryotic cells. Right. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasmas, PPLO. Basically, the monerans, you can see all the monerans, right? Only the monera group is prokaryotic. They are great. They are generally smaller and multiply more rapidly than the eukaryotic cells. To be noted, because prokaryotic cells, there's no membrane. There's no much complication, so it can they're much more smaller and they can multiply more rapidly as compared to the eukaryotic cells. Okay, so they may vary greatly in the shape and the size. So basically, the four shapes of bacteria is given: bacillus or bacilli rod like, coccus, cocci spherical, vibrio vibrim comma shaped, and spiral is spirala, spiralum, spiral, right? The organization of the prokaryotic cells is fundamentally similar. Prokaryotes are fundamentally similar, even though the prokaryotes exhibit a wide variety of shape and function. If shape and function varies also, the basic thing, like they are all having membraneless nucleus, and they have no membrane bound organelle, like that. Okay. All prokaryotes have a cell wall. You can find cell wall. See, cell wall is missing only in which group of organisms? It was only in the Animalia part. But then, if you're talking about one exception, then it will be Mycoplasma. In Mycoplasma, there is no cell wall. Okay. The fluid matrix filling the cell is the cytoplasm, right? What is the main arena of all the cellularic activities? It is also the cytoplasm. There is no well defined nucleus. The genetic material is basically naked. Genetic material is naked, not enveloped by a nuclear membrane. There's no nuclear membrane, right? In addition to the genomic DNA, there's a single chromosome circular DNA double stranded. Okay, many bacteria have small circular DNA outside the genomic DNA. Okay, so these are called as plasmid DNA. These smaller DNAs are called plasmids, right? The plasmid DNA confers certain unique phenotypic characters to such bacteria. So there are two roles of it. First role is to resist the antibiotics. This is called as resistance factor. Okay, and later, yeah, DNA is also used to monitor the bacterial transformations with the foreign DNA. Whenever it comes in contact with the foreign DNA, what kind of bacterial transformations are there? Nuclear membrane is Yeah. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cells except for ribosomes, right? Prokaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane is called mesosomes. Is the characteristic of prokaryotes. They are essentially the infoldings of the cell membrane. Talking about the cell membrane, the infoldings of the cell membrane is called as mesosomes. To be noted that they are found only in the prokaryotes. Okay. Now again, another set of dimensions are given. A typical bacteria 1 to 2 micrometers. Pleuropneumonia like organisms 0.1 micrometers. Viruses and a typical eukaryotic cell, right? So You can remember from the smallest to the largest viruses 0.02 to zero point 0.2 micrometers. Then zero point 0.2 is done, zero point 0.1 micrometers, and then zero point 0.1, right? So starting from 1 to 2 micrometers, and 10 to 20 micrometers. Cell envelope and its modifications. Let's move to it. Okay. Yeah. How about yellow? Cell envelope and its modifications. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope, right? It's a chemically complex cell envelope. The cell envelope consists of tightly bound three-layered structures, which is the outermost glycocalyx, followed by the cell wall, and then there's the plasma membrane. Okay, so glycocalyx, cell wall, plasma membrane. So although each of the envelope, each layer performs different functions, they act as a single protective unit. As a whole, it's for protection, in spite of having different functions independently. Bacteria can be classified into two groups based on their differences in the cell envelopes and the manner they respond to gram staining, right? It's called the gram positive when they take up the gram stain, and it's called gram negative bacteria when they don't take up the stain. Okay. Glycocalyx differs in composition and thickness. Speaking about the outermost layer, glycocalyx, they differ in their composition and thickness among different bacteria. It could be a loose sheath like a slime layer. Slime you would have seen, right? It's loose, and in capsule, it's thick and tough. The cell wall determines the shape, it gives a structural support and prevents the bacterium from bursting or collapsing. The usual work. Hmm. They also help in cell to cell interactions. They prevent the entry of any, you know, um, undesirable particles into the cell, right? Yeah. So plasma membrane is selectively permeable. It does not allow all the substances into the cell, okay, and interacts with the outside world. It's the plasma membrane which is going to interact with the outside world. This membrane is similar structurally to that of the eukaryotes, okay. Prokaryotes are fundamentally similar to each other, number one. Now, towards plasma membrane is, basically it's the same in both prokaryotes and eukaryotes. Okay. Speaking about the special membranous structures, it is called the mesosomes, already spoken, right? They are the inclusions which are found only in the prokaryotes. Okay. These extensions are in the form of vesicles, tubules, and lamellae. They help in cell. Yeah, this is important. You can remember in a flow cell wall formation, DNA replication, distribution of it to the daughter cells. They also help in respiration, secretion processes, increasing the surface area of plasma membrane as well as the enzymatic content. Okay, and also it was assumed, or it is the mitochondria of of a prokaryotic cell. Okay, mitochondria are usually helping in production of energy, respiration, and all of that, because in prokaryotes there is no membrane bound organelle, there's no mitochondria as well. In spite of that, or in place of that, we have mesosomes which help in respiration. In some prokaryotes like cyanobacteria, blue-green algae, okay, other membranous extensions. Other membranous extensions in the cytoplasm called as chromatophores, which contain pigment. It's the same as mesosomes. We're calling it as chromatophores because they contain certain pigments. Okay, bacterial cells may be motile, may be non-motile. Yeah, if motile, they'll have a thin filamentous extension from their cell wall, okay, which is called as the flagella. And bacteria shows a range in the number and arrangement of flagella. So you can find that they have filament hook in the basal body, parts of a flagellum. The filament is the longest portion, filament, okay, and extends from the cell surface to the outside. So what are the three parts of a bacterial flagella? Basal body, hook, and the filament, right? Yeah. Besides flagella, pili and fimbriae are also surface structures of the bacteria that do not play any role in motility. Pili and fimbriae, no role in motility, right? But the pili, which is an elongated tubular structure which is made up of a special protein, and fimbriae are small bristle-like structures which are sprouting out of the cell in some bacteria. So if there's no role in motility, what is the point of having it? It has two roles. It attaches the bacteria to rocks in the streams, moving streams, so it should attach, right? Yeah. And also to the host tissues. When they enter into the host, they have to, attach to the tissues. So that's the point. Yeah. Moving to the ribosomes and the inclusion bodies. In prokaryotes, ribosomes are associated. Yes, ribosomes are associated with plasma membrane in prokaryotes. Important of the cell. They're 15 nanometers by 20 nanometers in size. They're made up of two subunits. See, basically, ribosomes can be 70S type or 80S type. S is the sedimentation coefficient or Swedberg's constant. 70S has two subtypes and 80S has two subtypes, 50-30, 60-40. Remember, they're not additive. 50 plus 30 is not 70, of course, but then they are the subunits. Okay, so this, and also in prokaryotes, there is only 70S type, but in eukaryotes, you can find both 70S and 80S. Okay, yeah. So now they have two subunits, right? This 50S, 30S, and 60-40. Okay, so they are joined together by magnesium ions, Mg2 plus ion. We'll be studying it later again in the same chapter. Okay, yeah. So now, hmm, ribosomes are the site of protein synthesis. They involve in protein synthesis. Now, this is important. Several ribosomes may attach to a single mRNA and they form a chain of, it's called as polyribosomes, polysomes, or even ergosomes. Okay, so the ribosomes help in, yeah. So the ribosomes of a polysome translate the mRNA into proteins. In the central dogma, it is seen transcription and translation, right? So DNA to mRNA, mRNA to protein. So that second step, mRNA to protein, which is called as translation, is by the polyribosomes or the polysomes. Okay. Speaking about the inclusion bodies, they are the reserve materials in prokaryotes are stored in the cytoplasm in the form of inclusion bodies. They're not membrane bound by any membrane system. So basically, we are talking about prokaryotes. So of course, there is no membrane over here, and they just lie free in the cytoplasm. You can find three types of inclusion bodies: phosphate granules, synophycean granules, and glycogen granules. Phosphate, synophycean, glycogen. And there are gas vacuoles, which are found in blue, green, and purple and green photosynthetic bacteria. What is the role of this gas vacuoles? It helps in buoyancy, floating, right? It helps it helps the bacteria to float, right? Moving to the eukaryotic cell, right? Yeah. In eukaryotes, which includes protista, animalia, fungi, and plantae, except for monerins. In eukaryotic cells, there is an extensive compartmentalization of the cytoplasm through the presence of membrane-bound organelles. Basically, they have membrane-bound organelles, because of which you can see compartmentalization means there are certain compartments like mitochondria is here, you can find Golgi over here, ER over here, like that. Eukaryotic cells possesses an organized nucleus with nuclear envelope. In addition, they have a variety of complex locomotory and cytoskeletal structures. It's not that simple like our prokaryotes, right? Yeah. So the genetic material is organized into chromosomes. It's not lying freely as in the prokaryotes, but it's, genetic materials are organized into the chromosomes. All eukaryotic cells are not identical. Are all prokaryotic cells identical fundamentally? Yeah, basic ways they are identical, right? Yeah. Plant and animal cells are different as the former. If speaking about the plant, they have cell wall, correct? They have plastids, chloroplast. Animal cells don't have that. Of course, plastids have three types, but just speaking about the chloroplast. And they have a large central vacuole to be noted. In both, they have central vacuole, but they have a vacuole. But plant, it's like about 90% of the plant cell is the vacuole. Okay. Yeah. And talking about the animal cells, they don't have all of these. Of course, they have a vacuole, but it's not that large. And also, plants, animal cells have centrioles, which help in cell division. It's not there in plant cells, right? And is centrioles membrane bound or not? It's not membranous, right? Yeah. So these diagrams and the further part will be done in next video. Until then, definitely spread the key of success, spread knowledge. Thank you.